Welcome to the Amphibian Development module. In this talk, we're going to learn about fertilization. So let us first look at what are chordates. So organisms belonging to the phylum of Chordata are chordates. These include the subphyla of Vertebrata, from which humans are, Tunicata, and Cephalochordata. All of the organisms belonging to the phylum Chordata have a notochord and a dorsal neural tube during at least some stages of their lives, if not all stages. The notochord is a stiff rod of cartilage, while the dorsal neural tube forms the spinal cord in the case of vertebrates. Now, some chordates, like tunicates, and we've seen about tunicates in a few chapters before, show the formation of a notochord and a dorsal neural tube during embryonic development. However, these structures degenerate during metamorphosis, and hence these chordates don't have a spinal cord, and hence they are invertebrate chordates as adults. Since the tunicates do form a notochord and a dorsal neural tube during embryonic development, we can actually say that we are related to tunicates because we also go through similar types of developmental processes. All chordates are deuterostomes, and thus all the organisms that come under the subphylum Vertebrata that include fish, amphibians, reptiles, birds, and mammals are also deuterostomes. Now, fish and amphibians are anamniotic vertebrates because their eggs do not have a structure called as the amnion. The amnion allows eggs to develop in dry land, and thus without an amnion, the fish and amphibian eggs cannot develop in dry land. Most fish and amphibians are oviparous. They tend to lay hundreds of eggs in water when they show external fertilization. So while many fish and amphibians show external fertilization, there are a few of them that also show internal fertilization. Thus, those organisms that exhibit external fertilization, the females will lay hundreds of eggs in water. Now, when we look at the development of amphibians and fish, they do share processes and genes that are used by other vertebrates like mammals. And thus, they are used as good models to study development and how body axis and organs are generated. Let us now focus on amphibians. And in this slide, we're going to learn about a little bit about the oogenesis in amphibians. Now, please note that this is extra information and is not present in your book. Now, in case of amphibians, the cells that will form the eggs are called as oogonia, and they become primary oocytes during the process of development. Now, oocytes tend to grow in size and store a lot of ribosomes and RNA so that developmental processes can occur once fertilization happens. They also acquire yolk proteins, and these yolk proteins are not made in the oocyte but are actually made in the liver, and the oocyte absorbs them through the bloodstream. Now, as the oocyte matures, pigments are deposited in the animal hemisphere, and thus when we look at the picture, we can see in the upper corner of the picture where we can see the developing oocyte, half of the oocyte has a dark color and the other half of the egg is light color. The dark part is on the top part, which is called as the animal hemisphere, whereas the bottom part where we see more of the yolk present will be called as the vegetal hemisphere. In the case of frogs, fertilization tends to be external. However, if we consider another group of amphibians, which are the newts, their fertilization tends to be internal. In this talk, let us focus about the fertilization in frogs. Now, male frogs fertilize the eggs as the female lays them, and that is why fertilization is external. The frog egg has a polarity before fertilization because it contains proteins and messenger RNA in specific places. These proteins and messenger RNA are maternal proteins and maternal messenger RNA. Frog eggs have dense yolk that is present at the vegetal end. On the other hand, when we look at the animal hemisphere, it is darker in color due to the pigmentation of the egg, whereas the vegetal hemisphere tends to be lighter in color. The cortical cytoplasm in the animal hemisphere is pigmented, which also contributes to the dark color. Fertilization occurs in the animal hemisphere, and the point of sperm entry is able to determine the dorsal ventral axis of the organism. The point of sperm entry will form the ventral side of the developing organism, while the part of the egg that is about 180 degrees opposite to the sperm entry becomes the dorsal side of the developing organism. After the sperm enters the egg, the centriole of the sperm organizes the microtubules to form these parallel arrays that are able to make contact with the opposite end of the egg. These microtubules allow the cortical cytoplasm to rotate by about 30 degrees with respect to the internal cytoplasm. So as we see in the figure, the dark gray pigmented cortical cytoplasm has now rotated. However, the light gray diffuse black pigment has not undergone that rotation. This is called as cortical rotation. In the case of some amphibian eggs, this movement of that pigmented cortical cytoplasm results in the formation of a structure called as the gray crescent. So when we look at the figure, we are able to see an egg that has not undergone fertilization or cortical rotation. However, in the figure to the right, when we can notice where that blue box is, we can see the formation of that gray crescent. The gray crescent is known to play important roles in developmental processes because it has different morphogenetic determinants. It is also now known that the gastrulation occurs in the area where the gray crescent is formed. Let us now look at the role of gray crescent in development and how embryologists determine the importance of the gray crescent. If an experiment was performed where we took a fertilized egg and allowed the first cleavage event to occur in such a way that both daughter cells had the gray crescent equally divided among them, and then we separated those two daughter cells or blastomeres and followed their development, then what was found was that each individual blastomere was able to give rise to normal larvae. And thus they had all the morphogenetic determinants that was needed to give rise to the complete organism. On the other hand, if another experiment was performed where the first cleavage event occurred in such a way that only one daughter cell had the gray crescent while the other one did not, and then when we separated these daughter cells and followed their development, then it was found that the blastomere or the daughter cell that had the gray crescent was able to form a normal larva. However, the other daughter cell that did not have the gray crescent was unable to form a normal larva and instead formed a piece of tissue which was named as the belly piece. These experiments showed that the gray crescent has morphogenetic determinants that are crucial for development. With this, we come to the end of our talk, where we learned about chordates and how fertilization occurs in amphibians. We also learned about cortical rotation, which occurs after fertilization happens and results in the formation of the gray crescent, which is visible in some amphibian eggs.